Good morning, everyone. First of all, I would like to take this opportunity to thank particularly the Department of Crop Physiology at TNAU for this opportunity. And today, I'm talking from wild to refine, maximizing genetic gains for industrial traits and environmental adaptation. Before I move forward, I would like to acknowledge that Agriculture Agri-Food Canada Research Farm is located on the Treaty 4 territory and the homeland of Métis. We pay our respect to the First Nation ancestors of this place and reaffirm our relationship with one another. We would also like to recognize that some of you may be attending this event from other indigenous traditional lands. In doing so, we are actively participating in reconciliation as we navigate our time in learning and supporting. To get going, let me introduce you all to poplar species, one of the fast growing trees in the northern hemisphere. It is very easy to propagate, similar to either your sugarcane production systems using the cuttings or the mulberry production systems. And it is widely distributed throughout the northern hemisphere. And it is dioecious, where the male and female are two different trees altogether. So in a lot of other crops, you see male and female in single flower or in two different floral structure, for example, corn, tassel, and silk. And it produces flower at the age 8 to 10 years. So until that, you don't know whether it is a male tree or female tree. And the genome is relatively small, around 550 megabase pairs. When we look at the distribution range of these cottonwoods across North America, one can see the brown area is Populus balsamifera that goes from all the way from the Alaska to the west to the Atlantic Ocean in the east. And the green one is the Populus trichocarpa, another species of Populus. Whereas when you see the pink one, that's one of the drought hardy Populus occidentalis. And the other blue one is Populus occidentalis deltoides. So there are several species and subspecies. When we look at the tree breeding program at Agriculture Agri-Food Canada, it's almost 120 years old. If I call myself the fifth generation fighter jet, I am the fifth generation breeder for AFC, where I took over in 2010 at the end of my PhD for my breeding program. Over these 100 years of efforts have led to conservation and development of 15,000 unique poplar germplasm in Canada. Our goal in the breeding program has been that we want to use the native species, particularly as poplars become multifunctional crop. Our program focus follows selection of superior genotypes from genetically diverse base population of native genotypes with the aim to develop feedstock that can deliver not only the environmental benefits, but also provide renewable energy. This led to the establishment of Agriculture Agri Canada Balsam Poplar Collection, commonly referred to as AgCan-BAP. What I meant by AgCan-BAP is like when you saw the previous slide, I showed that brown map. Now it is a much closer map of the species range. It has almost east to west 8,500 kilometers length, and north to south is almost 3,000 kilometers. So it's almost the size of India. We were able to go and collect the germplasm throughout the species natural ranges. Each of these green dot represents a population where within a 30-kilometer radius, we were able to collect 25 trees altogether. And each of these trees, like as we go further north, the day length, where here in Tamil Nadu today, it may be very close to like 11 hours or 10.5 hours of day length. There we get up to 24 hours of sunlight. The sun never sets down. Whereas the southern at the south, it gets up to 16 hours of day length. So you go from 16 hours, as you go further north, you go up to 24 hours. Whereas seasons up north are roughly 2 months of growing season. The plants only grow for 2 months, then it gets covered by snow. As we come south, the plants in the east to the far of my slide, they grow up to 6 months. So you go from 2 months of growth to 6 months of growth, depending upon where you are growing the trees. And each of these stars represent the species natural range. For example, here, this is like where my breeding station is located, called Indian Head. And we moved the trees 450 kilometers north to understand what happens with climate change if trees move north. And if trees moves outside species range, what happens to them? And also, if trees moves into drier climate to more wetter climate, what happens? And also, if trees move furthest north, then we put them in Alaska and understand how those trees behave. From each of these locations, we collected trees. The cuttings were planted in individual gardens at all these locations. We also planted one location further south. Why trees should always move north? What happens if we move them further south during climate change? So, you making use of all these multi-location trials, we wanted to understand how trees respond to climate change that is happening rapidly at northern latitudes. So what we did was we went into the forest areas during the month of January to March when Canada is covered by snow. All the white stuff you see here is snow. So we went into forest, we cut these branches, we bring them and we cut each of these cuttings and we grow them into individual ramets or clones. So each tree will become its own clone and we planted them all into different sites that I showed in my previous slide in the stars. So they were all planted into different common gardens. Now they are almost like 14-year-old tree genetics. So in the interest of time, today I focus my talk just on the Indian head, which is the southern fringe of the species range, and one outside the species range. What happens if all the 1,000 trees that we collected throughout the species range are planted in this location and this location? So what's the difference between these two locations? When we look at Vancouver, which is this site, the seasons are long because it's very close to Pacific Ocean. The seasons are very long and photo period, the day length during summer is 16 hours, 5 minutes and temperatures during summer are around 20.5 degrees Celsius. When we look at Indian head, which is the southern fringe of species range, the growing season is very short. Climates are more continental because you move away from ocean. And here the temperature is almost 1 degree warm. Once we planted all these trees into these two sites, what we did was when the snow melts, the bud will be like this. When the snow melts, temperatures increase in northern hemisphere. The leaves, the buds flush, the new leaves emerge. They continue to grow in height. Then they tend to cease height and set terminal bud. So we were able to monitor year after year when the leaves come out, when the leaves stop growing in height. And then we were more interested in when the leaves change color, going from green plant all the way to leafless. And when they change 50% yellowing to 25% yellowing, 50% yellowing, 100% leaf drop. So you look at calendar date, you say, okay, June 20th it flushed, September 20th it changed color. You monitor year after year what happens to these trees when you plant them in two different environments. So what we are trying to understand is what genetics does under a given environment. So here I'm showing latitude where the trees were collected from, whether we collected at southern latitude or in Arctic zone and all. And on y-axis, I'm showing calendar date. 60 is March 1st, 120 is March 30th, like you go day 1, January 1st to 365 days, December 31st. So I'm showing the Julian calendar. And here I'm showing the two sites where the red is Indian head that has short growing seasons. Vancouver has long growing seasons. They come out the leaves come out very early there. And the difference between these two common gardens is almost 40-day delay when the leaves come out. When we look at the same for when the buds are formed so that the plant stops growing in height, these two lines converge. The difference is now just around 20 days. And Some of the trees stop growing from high latitudes much early before the summer even begins in Vancouver. So we were very much interested what happens to these trees when summer comes way early. So height growth cessation is manifested by formation of the terminal bud that you saw on one of the slides. Is primarily controlled by day length because here at this latitude 65 to 70 degree, it's always 24-hour light. But at my common garden where I have planted these trees, it never gets to 24-hour day length. The maximum is 16 hours. Plant always thinks winter is coming, I have to get ready for winter. So they stop growing. When even though you give good temperature, they are all hardwired that, oh, winter is coming, I should stop growing, get ready for winter. Then when the leaves change 50% aloneness, both the lines converge. The reason for that is when we take September 21st, no matter whether you are in Coimbatore, whether you are in Vancouver, or whether you are in Arctic, it's all 12 hours day length. Similar March 21st, we all have the same day length, whether you are at equator or whether you are in Arctic. So plants always say, okay, I have lost my safety net, 12 hours day length. Then they all, no matter where they come, they all tend to change leaf color and try to drop their leaves off so that they get ready for winter. In that observation, what we were interested is what happens to the green cover period, even though the temperatures are warmer, why the plants are not growing taller. That was our question. So we wanted to understand the leaves should accomplish more photosynthesis. That means it should result in taller plants. What were growing for 2 months, we gave them 6 months of good ideal growth, but they didn't grow tall plants. So other way is what happens to high latitude trees when spring arrives early because of climate change? You can ask the same question in a different way. But here I'm showing the solar calendar going from January 1st to December 31st, 365 days, how the day length changes in Northern Hemisphere. On June 21st, it gets the highest day length. On December 31st, January 6th, we get the lowest. So this is always fixed for a given location. This is Fort McMurray, where June 21st, it gets 18 hours of and 3 minutes of sunlight. That's always fixed, no matter what happens to climate change. The sun length don't change. The same plants at Fort McMurray, they sense the 16-hour on August 6th, and they say, oh, winter is coming, I have to get ready for winter. As you wear sweater, the plant tend to shed leaves and it get ready, putting all its carbohydrates into the bark and get ready for winter, what we call sugar osmolytes. That's what gives cold tolerance, similar to drought tolerance. So today, the leaves come out, buds flush on May 30th. They grow all that much. They stop growing here and they change the leaf color, drop their leaf, get ready for winter. If spring comes 20 days early, the plants definitely take advantage of that. They grow taller. Spring comes much advanced. They take advantage of all these 40 days plus this much growth. That's why with climate change, people say we may get taller trees, we may get better crops and all. But if spring comes much early, March 30th, when the leaves flush in a given location much early, this is where the problem is. Instead of sensing this day length of 16 hours in September, they sense this in May itself. Then they say winter is coming. As soon as they see that absolute day length, they think, oh, winter is coming, I have to stop growing. That's what happened for a lot of northern trees when spring arrived early in Vancouver. So the spring shoots that flush here, the leaves become big enough to sense day length. Then they are physiologically ripe to respond to critical day length upon gaining competency. They set bud and fail to utilize the best part of summer. That's what happened. This is experiment. I will show how we did it. Then, so here I was showing 35 days is the minimum growth that is required for plant to understand its surrounding better. So now I ran that This is the climate normals. Today, at that location, we get 59 days of growth, where the plants flush on May 6th, they gain competency, summer height growth ceases on July 14th, August 7th, they go dormant, and 60 days is the growth. Okay.
Shallow nest or scrap nest.
Sir,
So when I ran all the three models, here I have taken RCP 4.5, which is a mid-level climate change model. Under certain scenarios, when the climate comes very extreme, April 8th, they flush, they gain competency by May 8th, and June 5th, they go dormant. That's very similar to what we were observing in the field locations. So in that scenario, how one can avoid the mismatch between photoperiod, which is fixed no matter what happens to climate change, phenology, which is a highly heritable trait, and physiology. So we went on to develop a new hypothesis called the phenological mismatch may be avoided by moving photoperiodically appropriate genotypes along climatic lines that have east-west orientation. When you go around the globe, if you take this as globe, you put a point and you keep rotating, your day length is fixed. You go to equator, your day length is fixed no matter where you go around the globe, southern hemisphere, northern hemisphere. But if you go from equator to north, the photoperiod changes. But at a given one, it remains 12 hours, 13 hours. So we said, no, you move trees only along east-west orientation. So in doing so, you just not move a gene or a genes, set of genes, you can move the entire genome. Like poplar has 40,000 genes, and we said this is the best way to move all the genes to a new climate. What I meant by that was, instead of moving the trees from southern trees to north as climate changes, you don't do that, but you move them with the east-west orientation or west-east orientation, where you move them at 16-hour day length, 17-hour day length, 18-hour day length, 24-hour day length. Then you are not affecting the tree to understand its day length. So we published this work in early 2009. Then in 8 years later, Nature and Science mentioned trees in eastern U.S. head west, and American trees are moving west. No one knows why. And all they could explain is 20%, because the remaining 80% was the heritable trait that is controlled by photoperiod or day length. So we were able to show that this is the new way to move tree genetics outside the species range. When we look at the trees in our common garden, that is when we planted them outdoor, the local trees are the tallest trees that come from south, whereas the tree that comes from north, since it always are exposed to 24-hour, here it gets 16-hour light, but it gets 6 months of growing opportunity with warm temperatures. It never grows tall. It always remains the shortest when we plant them here. Local trees know that they can grow up to certain stage, they stop growing and they are taller. If that's the case, if photoperiod is driving tree growth, then it's very easy. You have built all this controlled growth chamber. All you have to turn the light to 21 hours instead of 16. That's all I did. I brought the cuttings, I grew them at 21-hour day length. Now you see the trees that come from very high latitude, they grow very fast. But since they were not exposed to 21-hour, 24-hour day length, they were not growing fast. So the southern trees grow slow, but they grow for very long period. These trees grow 2 months, they grow super fast. They are sprinters. Whereas these guys are marathon runners. They grow slow, but they grow long periods. That's how they save energy. That's the energy balance comes into picture. Then automatically the same experiment I ran in greenhouse, my experiment has reverse rankings, where northern trees are tall now, southern trees are short. So if you ran a greenhouse experiment, someone ran a field experiment, your results are exactly opposite. Sometimes people may say, oh, there is something wrong. No, both are right experiments. This was a clear demonstration to show that day length is very important for trees to adapt to climate change. Then the next question was, why trees in north at high latitudes grow really fast compared to the trees in south? So for that, we applied the physiological tools as you are all familiar. Here the CO2 moves through stomata, what we call stomatal conductance. And from the substomatal cavity, it moves to the site of photosynthesis to the rubisco area, what we call conductance. And then they convert it into photosynthates or sugars. And finally, it goes to the sinks, whether it is roots, leaves, stem, any that gets translocated from the leaf. We were interested, cut open the leaf, just like you cut open under microscope. We observed northern trees have more mesophyll surface area. The substomatal area is very large, whereas southern trees, they have much smaller area compared within a given leaf section when we cut open. Then the gradient for stomatal conductance and mesophyll conductance is very high. When we look at northern trees, they have much higher stomatal conductance and higher mesophyll conductance. We can see big differences in photosynthesis due to the gradient for CO2 to be drawn to the substomatal to the site of carboxylation is very steep. So the CO2 is always pumped much faster in north compared to the southern trees. Similarly, when we look at the trade-offs in disease resistances and carbon gain, how fast they grow, the trees from north here, 65 to 70 degree, grow really fast, but they are very susceptible to diseases because evolutionarily at that high climates, disease pressure is relatively low. Whenever we expose in southern gardens, they get very susceptible. So we were able to show the resistance for disease is very less, whereas in south, they are much more resistant. So the selective pressure for leaf pathogen is likely higher in southern parts as demonstrated here, and a higher copy number variation involving genes of defense responses enable resistance or heavier pathogen load. So that was what was giving resistances. Such scenarios, as a breeder, how I can exploit what we observed in nature instead of we trying to GM trees or develop other molecular method based trees by inserting a gene or set of genes. We were very much interested how I can learn nature and how I can exploit nature for my advantage. So far, what we learned was the trees that come from north grow really fast. They have higher photosynthetic rate. They are very cold adapted. They grow short season. They are very much susceptible to disease. And they have large mesophyll surface area. That's what we understand up to now. And in the south, it's exactly opposite. They have lower photosynthetic area, warm adapted, long growing season, small mesophyll surface area, and disease resistance. As a breeder, can I combine these traits and take advantage? What's good in both locations? Can I exploit that in my breeding? So the desirable traits can be combined in a progeny by exploiting natural trait diversity for environmental and industrial applications. So what I meant by that is adaptive trait-based tree deployment is in one breeding program, we had a very ambitious plan, one breeder, one breeding program. We should be able to select the right tree for right climate. If you consider the entire landscape of India, my breeding program deploys trees for the entire country. The right tree for the right climate. What I meant by the right tree for the right climate is the tree you plant today, 2023, it will stay for 100 years. That is 2123. The climate of that place would change gradually. In 100 years, the temperature, rainfall, everything at Coimbatore will change. You have to design a tree for that climate today. You can't change the trees that day. Oh, I will change that day. That's not possible. You have to think today and develop and deploy the right tree, understanding what would be the climate of Coimbatore in 2123 today. What I meant by that is these trees stop growing at 200 days on the calendar date, but they are fast growing. So I can select these trees and deploy at very high latitudes where sunlight is 22 hours during summer. Whereas at 49-degree, very close to US border, these trees grow much faster. I can take those trees and deploy for 16-hour day length, and in between I can select for 15 to 19-hour day length these trees. So for a given, I can select the fast and best growing trees. Can we do that really? So here I'm showing we were able to exploit natural traits. These are the very weak trees that are very low in lignin. For biofuels, to make ethanol, you need to break down lignin, which is industrially very expensive process. So by having the low lignin fragile trees, we don't have to do any selection. I can go to the garden and tell my technicians, stack these 5 trees, they are low lignin types, and then we can mass produce them. Compared to the trees that are standing very straight, they are very hard, high in lignin, they don't bend much, but these produce the best ethanol for us versus these ones. So we were able to just exploit from the nature the low lignin types. Now let me introduce you to one of the ongoing project that recently ended called PopCan project. Here, as a federal breeder, I sit here and I provide all the 1,000 trees to different traits, whether it is metabolomics people, physiology people, pest and disease entomologist, pathologist, then wood chemists and phenologists. All they characterize all these trees, the same set of trees for every department comes and works on the same set of trees. Then the same trees goes for genomics people, transcriptome people, methylome people. They sequence all the trees and they submit all their data to the SNP database, and all these people submit their disease resistance, pest resistance, physiology, metabolome, sugars, everything to the trait database. And the computational biologists, climate modelers, they all utilize this data, bioinformaticians, and they curate the data and they make it that available for comparative genomics and population genetics. And these two people interact and they feed me what are the right trees to plant and how. And my role is to oversee all these operational activities and also deal with government on policy, regulations, and also commercialization of these trees for farmers or for other industrial applications. So the first question we asked in this project is, can I move all 40,000 genes to new climate? That means I can take a tree, can I move that tree to a new climate? I'm not moving a gene or 2 genes or a set of genes, but I'm moving the entire genome. Can we do that? So to do that, one can harness the standing adaptive variation for climate-driven traits. What I meant by that is we have genotype. We have a given environment where the trees we collected from different locations. And we have phenotype. They have their own physiology growth. They have their own stomatal numbers, photosynthesis, and everything. Here, what we did was we sequenced all the 1,000 genomes and we did 24 different species assembly, and we did 350 bisulfide epigenetic sequences, and we characterized 99 traits for every tree in every climate. Phenology, growth physiology, biomass, wood characteristics, leaf morphology, metabolites, disease, wax, we characterized. And we did the geospatial modeling. That means we took the current climate for the given location and we applied future climate models. And we were able to use climate SNP variation and we made all the data public. So if any of you are interested for your master's PhD to use our data and do your own thesis, you are most welcome to do that. Data is publicly available for anyone to use. And we give to, you have new idea, new way to analyze this data, you are welcome to do so. What I meant by the genotype environment is, you might have heard the word GWAS, Genome Association Studies, where you take the genotype and phenotype and you run a linear statistical model and you understand how a certain SNP or a certain gene is related to a given trait, whether it is related to water use efficiency or disease resistance. And here we have physiology, environment, and phenotype, where you have built the growth chambers. It can be warm temperature, cold temperature, less water, high water. you can learn ecophysiology. What we started to understand was how the genotype and environment are related. A genome is fixed, environment is very dynamic, it changes. And we went on to provide the first experimental evaluation of ability of what we call genetic offset or genomic offset, referred to as genomic vulnerabilities, a matrix of climate maladaptation to predict organism response to climate change. So we were the first ones to show whether it is for animals, humans, or for plants, how one can calculate the vulnerabilities of the genome for the future climate. So let me walk you through last 20,000 years, what happened in Canada. When you look at the top panel, this picture, this is 21 years ago. Canada was covered by 1 kilometer thick ice. So if you stand on ground and you put 1 kilometer thick ice, that was what was covered 21,000 years ago. 16,000 years ago, some of that snow on the southern ranges. 13,000 years ago, this ice sheet opened up, it broke into two pieces, and plants were able to move. The seeds, all the seed transfer could happen through wind and everything through this ice opening. Then 11,000 years ago, ice receded further northwards, pole wide, and currently we see the range of populous species, what we see today. So it took 20,000 of climate change to happen. What we are experiencing today is much rapid climate change. We don't have 20,000 years to adjust. We have to adjust in 200 years. So we have to think different about trees and tree generation. All the data to show like what has happened in those time when the sudden ice age came. We can see fossil records of dinosaurs, trees all submerged underneath. And when we do carbon dating, we can understand when these trees kind of got killed or dinosaurs got killed. So, and the ice was covered. When we do some of the course through Arctic and others, we can understand how old the ice is. Yeah. And today, when we see the population structure of this balsam poplar, during ice age, they took refugia in US. As the ice receded northwards or polewards, they moved to Alaska and some moved to the east. And that's what happened. Ancestral was the central population, they moved east and north. And it was the migration. The seeds fell this year, they germinated next year, then the new trees grew, then they germinated. Continuously, they kept moving northward. It was not the mutation that led to the northward movement. It was the massive historical migration of the seed that led to the colonization of the new climates. When I take a single gene, you might have heard the circadian clock. Whether you take any crop, here we have Gigantia gene, FLC flowering gene, and FT flowering locus, and LHY, TOC, all those genes you might have read in your MSc Masters. Today I'm just showing this gene Gigantia, and that's how that gene like look. Add another 39,999 genes, the genome gets very complex, and the landscape, what you see, is very diverse. Next, we were interested because, as I said in my first or second slide, that these are male and female trees. As a breeder, I don't know until age 10 whether the tree is male, whether the tree is female. So I told my colleagues that, hey guys, we need to identify the marker to know whether it is a male tree or female tree before at day 5, day 6. So we were able to like run the GWAS, and what we identified was, I'm good. So we were able to look at this ARR17 gene, which is right here, that showed the highest association and a strong predictor of gender. And we also found that as humans, males and females have different strengths. Whereas when it comes to trees, being a male tree or female tree, there is no ecological advantage. Except you bear pollen, you bear seed, there is no other advantage. Whereas sometimes we feel women are much weaker. When we tested that in our trees, we didn't see anyone because it's a single gene. Our evolution never allowed that gene to be very aggressive or very dominating in either of the sexes. So here, when we look at the methylation, the gene is present, but it never get expressed. It is suppressed always until the age 10, very similar to our genders, where certain heritable traits or certain features are suppressed. Once they reach puberty, it get expressed. It's very similar in trees. They are suppressed through epigenetic signals. As you can see in the first intron, the male and female are differentiated, whereas at fourth intron, you don't see any differences. So we were able to show that male and female trees, they diverged just 7 million years ago. That led to the differences in having like a pollen can be transferred onto 10 females. That means you can have 10 half sips where a single father can pollinate 10, or a mother plant can receive 100 pollen. Then every seed is different. It has its own half fixed mother, but the fathers can be 10 different fathers because the pollen can be from any source of father. That allows rapid diversity. If you have a male and female on the same, it gets self-pollinated. But what this allowed was it allowed for cross-pollination and increased diversity. So now moving to, you might have read or heard all about like artificial intelligence, machine learning, how we utilize machine learning, whether it is forestry or whether it's agriculture. Here we took all the snips and what we call like neural network analysis. We were able to do supervised neural network analysis where each of the snips with traits and they were sub-associated. Finally, you have xylem, you have leaf. And when we look at the leaf xylem, here the R square is 3.5, R square is 4. But when we combine some of these models, you get better prediction of not just the heritable traits, but also non-heritable traits as well. So the deep learning models offer new insights into epigenetic influence of plant traits that can be used in breeding to model phenotypes that are poorly estimated using genomic predictions. So how we utilize some of these mathematical tools or computational tools in our breeding. Why are we interested? By now you are thinking, who cares about trees? Why are people investing so much money and resources into tree breeding? For us, we define for remediation and efficient energy supply. As Canadian farmers become more concerned about land management, nutrient management, heating costs at farm level, these popular fast-growing trees emerge as multifunctional crops. When we look at the prairie climate in Canada, what we call the wheat bowl of the world, in early 1900, it was just grasslands where there were like no trees. Farmers had very hard time when the early settlers moved in. But today, 100 years of tree planting, you can see agroforestry, tree rows planted all across the farm that gives snow trap, that minimizes soil erosion, creates microclimate for crop production to happen in these fields. So that has led to realize the climate. The more likely, I won't say climate, but more of a microclimate. Similarly, these are potato fields. Farmers apply a lot amount of fertilizers, nitrogen, phosphorus, and all. Lot of that runoff happens if there is rainfall events. And we build buffer walls, riparian buffers, a 10-meter wide or 15-meter wide buffer strips all along the water bodies. So what happens is when the runoff of fertilizer happens during rainfall event, those nutrient is intercepted by these trees and you can go in the month of November, December around this time when the ground is frozen, snow covered, you harvest all the wood. Next year, they just grow back just like mulberry and you continue to harvest every 3-4 years. In doing so, you can 300 kilograms of nitrogen per kilometer buffer and 45 kilograms of. Otherwise, when you see algal bloom, that's due to fertilizer, particularly phosphorus runoff, and leads to algal bloom in freshwater bodies, whether it is your river systems, whether it is lakes, and all. So we were able to build all along water bodies that. And when we look at the piggery industry or dairy industry, they produce a lot of manure. How to deal with that manure. So we were able to apply all that manure to grow on marginal land. which is not productive for agriculture, but you can grow a lot of trees okay there. So we were able to use that marginal land, grow the trees, and we evaluate which trees take up a lot of that nutrient rich manure and use it for producing the best biomass or best trees. Similarly, we also have projects where we utilize whenever you go to toilet, you flush the toilet, the water comes out. That's water never get recycled. It just get recycled, then it get released into the systems. What we were doing was we took that water from the tertiary tanks before the municipality drains it into ditches, and we put them through pipelines. You can see here the pipelines running all along the length. We were able to irrigate that nutrient rich or somewhat nutrient rich water from tertiary tanks to grow biomass. And now big companies come and buy these acreages for as carbon credits. If they want to show that they're doing something for environment, they can buy the credits from city of Coimbatore. All the toilet water goes for growing trees. And for example, any Lakshmi Mills or Air India that carbon credits and show we are doing environmentally good work. So here we were working closely with SAS Water, just like Coimbatore Water or Chennai water systems. We work closely with them and how we manage the municipal wastewater for growing biomass. In addition, what to do with all the wood that you harvest? You can use it for furnitures like these plywoods, or else you can use on the farm. You can chip that wood, you can load it into this tank, and this is the bioburner we installed as demonstration to farmers where my greenhouses, office labs are all located. During the October, when temperature goes to -10, -15, all the way up to April, temperatures can go in Canada up to -30, below zero. So we were able to heat the buildings, greenhouse, office using the wood that comes from our research projects. And this costed like us $280,000, and it is a 1 million energy with 300 kilowatt, and it runs at 90% efficiency. So from October to April, I don't use natural gas or electricity to heat my buildings. It is the wood that we burn, and it is carbon neutral. Whatever you sequestered, you release only that much. Otherwise, if you take coal or petroleum to heat, releasing much more carbon footprint to the atmosphere. So, in summary, what we learned today was the relevance of population at extremes of species range as a possible reservoir of future climate adaptation. We also learned about our natural or artificial adaptation of trees to future climate is not just about temperature. When people talk climate change, they talk about two things: temperature, drought, heat, or CO2. But we never talk about the changing relationship between year to year consistency, that is day length. Day length is fixed no matter whether you go 2,000 years back or 2,000 years forward, it's fixed. Everything else changes around it. And plants are very, very smart. They never trust temperature at all in their life. They always believe in sunlight because it's fixed. The only thing temperature does, either it speeds up flowering or it delays flowering. Water, drought, it speeds up flowering or it delays flowering. If there is more rainfall, you have vegetative growth. If there is less rainfall, you have flowering. But plants always use safety mechanism, sunlight, whether to flower or not to flower. Because you take any agriculture crop, the plant is not producing yield for you. It's producing its own progeny. So it always uses day length as a safety mechanism, whether to produce seed, whether it can have. Us as scientists, we see plant as making food. Plant never do that. Plant produces its progeny, which is its seed. So lastly, a balanced coexistence of food and fuel has capabilities to deliver clean energy solutions while achieving environmental sustainability by involving farmers and isolated communities in any country's economic growth picture. With that, I acknowledge all my collaborators and different funders.
History alone, why you are focusing more in research in that area rather than any arrived species will be there, particularly what reason is science?
Yes. The question was, why are you focusing on this particular tree species, which is balsam poplar? Why not others? My breeding program focuses on 32 different species altogether. So poplar is one of these species that I today showed. We have berry-bearing uh, shrubs and trees, flower-bearing pollinator. For pollinator habitats, I breed for the shrubs to plant as agroforestry. Then for small birds to come and utilize that landscape, also breed for berry-bearing, fruit-bearing trees as well. So in that context, I use 32 species. In the interest of time, I just focused.
The popular trees,
Yeah.
whether it is applicable to other crops as well. For example, wheat or canola or lentil.
Yeah.
For the plant.
The question was, can we apply this photoperiod related thing to other crops? Yes, the answer is yes and no. Photoperiod sensitive crops, we have photoperiod insensitive crops. There we don't have to work on this model. Photoperiod insensitive crop you can plant at any latitude, in any, whether you plant in June planting or Kharif or Rabi, you get your yield. Whereas certain crops you can't plant in Rabi or Kharif, so those are photoperiod sensitive. So it applies to, but this concept is not well studied in crops. They believe more with temperature. So I've been, when I talk to, for example, agriculture breeders, whether it is, let's talk rice, cotton, they always talk with temperature. They never take into account the day length. Now we are telling a lot of breeding programs, you have to take photoperiod into your models, to train your models with photoperiod and use this. Now people are going back and they're telling like, okay, when we look at certain photoperiod sensitive, for example, when you do the rice seeding, I did this model for what we call photothermal quotient, where you take the day length, where you take the thermal units and you combine. When you combine this, you can arrive at photothermal quotient. It has photoperiod, it has thermal units or the heat units. So the Bayer was able to do, when they do the Rabi crop, in North India, it was flowering bit 30 days late. Then I applied that model and we were able to showcase them. And now Bayer uses some of those genes to improve to select for photoperiod insensitive rice varieties, no matter where you plant, whether you plant in Punjab or you plant in Coimbatore. When they say 45 days, 50 days flowering, you expect. So those are some of the advantages we have. So now slowly breeding programs are trying to understand how to exploit day length connections and relation length and depth.
What will that public database give access?
Okay. The question was for online users. I mentioned it, the public access of all our data, whether it's genomic data or physiology or other data, and how that can be used for Coimbatore tree breeding and others. My intent here was, let me clarify that whatever data we have done, whether it's genomics, physiology, phenology, metabolome, or any of the things, pathology, entomology, we have built a database public can download. Let's say you have a different way to think. Oh, I have a different idea. Let me run. Instead of you trying to get a project, write a project, find student, repeat all the work again, we said no. We have not left any much to be others. We are confident that if others have any good idea, they should just use our material, our genetics, our genomes, our traits, and run their analysis and publish. So we had, in that project, we had 17 postdocs. So each postdoc came from pathology, entomology department, soil science department, who does soil microbiome, genetics department, another came from breeding department. So we had 17 postdocs. Every postdoc was given a full-fledged access. This is the data. You go, you run your analysis. and you publish. In that way, we thought like, okay, this is good way to encourage others who may have an idea, but they don't have funding. We have everything available. We just hand over them. All they have to do is analyze. A new way of analysis might come. Then they might say, let me test that. Then the material is available. So students can, if they have an idea, they can come, let us know. It's just an MTA that we sign, very simple paperwork. Maybe within 15 days, everything get transferred to you from. You just have to give a one-pager abstract, what concept you are trying to do. The reason for that is if someone else is doing similar thing, then we say, Bhuminathan is already doing, no point. You repeat the same. If you come up with something different, then we say, okay, this don't conflict with Bhumi's work. You please hand over, take the material, you run your analysis. You don't have to give us authorship, anything. All you have to acknowledge is we received all the data and genetics from Agriculture, Agri-Food Canada. That's it. Even you don't have to mention our name. So we have made it very simple, very easy for public to think big and utilize already existing data. We always believe in creating data, but we, with machine learning algorithms, with big data analytics, now is the time not to create data, use someone's data and use it for your thesis work. In this university, you may have 100 years worth of data, climate data, or rice yield data for different districts, different taluks. You can collect all that 1950, what happened, 1960, what happened, how genetics change, how yields change, how climate change. You can run a lot of analysis without even stepping outside your office building. Everything is, data is there, but students are not extracting that to the advantage. They're all going and creating their own new data. So now is the time to what we call metadata analysis or big data analytics. So if you are good at some certain computational skills or certain codings, you can just sit on your computer and you can run your thesis in 6 months instead of 2 years. So that's also thesis. You can take Coimbatore district or Tamil Nadu state rice yield or cotton yield, and what was the rainfall patterns of that year and what was the temperature. You have now you have to use that data to predict whether the yields have increased, whether the yields have reduced, what happened with inputs. Was the yield due to breeders? Was the yield due to farmers applying more insecticide, more pesticide, or more nutrients? So we can sort out all that and tell the breeder, you have done good job or you haven't done a decent job. So this is the time to look at big data. That's why we wanted to have the data publicly available to. Well, we have certain genome combinations through our breeding, which can survive the climate of 2100. These trees live 150 years. So our goal is if we can develop this genomic vulnerabilities or genomic offset better using random forest models, we should be able to deploy the right trees for the right climate. So that's our goal. When I say right tree for right climate, we gave some of the trees. If you see the Greenland on the map, we were able to transfer some of the trees to. So what we did was the Greenland, as the ice is melting, they sent out a call for all poplar breeders in France, US, and Sweden, Canada. Hey, we need some trees to plant there. Our ice is melting and we need some trees. So a lot of breeders sent their favorite genetics. I didn't send my favorite genetics. I sent the right genetics for Greenland by moving the trees from northern Canada to Greenland. Just I moved west to east. And after 5 years, they sent us report. People from America sent trees upwards, French sent upwards, Swedes sent upwards. We moved only east-west. And they said only Canadian trees survive. Everyone immediately replied, oh, that's cold adapted. Then I explained why we sent those. Then everyone got surprised, like, oh, you are building a new model. And now if you go to Greenland, the first trees to grow there are Canadian trees that were moved with east-west orientation. So we have been successful that way. And the other paper that I showed, like both nature and science, We don't know why. And the trees were moving east-west, not south. As per people sometimes assume certain things, temperature changes, everything move northward. So we were able to disprove that. No, that's not how it would happen. And that's not the right way to move trees. This is the best way. East-west orientation or west-east orientation are the ways to move trees. It happens both ways. Sometimes the branches break due to ice. Then they land on ground, and in the spring they get stuck somewhere in soil and they propagate into a new tree. One method. The female trees produce almost like million seeds per season every spring. So those seeds can disperse almost 1,000 kilometers. If there is strong wind, a seed, very tiny seed, can be carried almost to 1,000, 2,000 kilometers in the air just by wind. And the seed falls there when the wind reduces, then it germinates. So you have a tree that has gone too far away. And since it's also pollen load is very high, the pollen can travel minimum 500 kilometers. So that's where the diversity the same. The genetics remain the same. When we planted them in the field, right? We cut the cuttings. The one of the method was the cuttings fall onto ground and they grow into new plant. That's all we did. So we didn't use the seed method because we don't know where the pollen came from, right? All our trees that we collected were anywhere between 60 to 80-year-old trees. So in doing so, we knew that this tree is exposed to 60, 70 years of native environment, whether it's 16-hour day length, 24-hour day length, and all. If the pollen grows too far, sometimes it is not viable, or the seed may not withstand within a given climate. If it goes 2,000 kilometers northward, it may grow, but it get killed by winter naturally. So the natural selection also happens after the germination happens. So that's why we selected a minimum 60 years is the requirement for a tree. Then we know that long enough in that climate. Yeah.
The study shows that irrespective of that temperature, photography play a major role as compared to temperature.
Yeah.
But Canada is a cool climate country, okay?
Yeah.
Where mineralization plays important. It's temperature.
Yeah.
But how come your study disproves that concept?
No, I never said vernalize. I didn't touch upon the vernalization part. I just said like the December when the snow period, then they flush again in spring in the month of May and all. Vernalization is very important. That's what I was showing the photoperiod clock. What you are doing, vernalization is you are giving the dormancy period. Dormancy don't have to be -10. It has to be very close to +4, +2. That's what happens in Kashmir area and all. So all you do is you put them in a fridge for +4 for 20 days, 25 days. If you put them for 5 days, it still germinate, but it takes 20 days to germinate. Whereas if you give 30-day germination, vernalization, it germinates in 2 days. So the minimum requirement, what I say is chilling needs. We also have photothermal needs. Plant also has chilling needs, which I didn't touch today. Yeah, we work on both. When I select the trees, we select for cold hardiness, we select for drought hardiness. If they don't meet these two traits, then we don't.
From one slide, important 130 days of early spring, right?
Yeah.
But all the literature says we can put able to get 1 or 2 days early spring, right?
Yeah.
But you got 130 days. So
Yeah.
that is one thing. So how it is naturally possible? Second thing, what happens to the pollinators because one has change in.
Yeah. So your question was related to the same amount of cold. So the spring arrives early, whereas here the spring arrives late. So that's why the trees there were able to get the heat units and they flushed. They met the chilling needs. They were waiting for temperature to warm up. As soon as temperature warmed up, they flushed. Whereas here the snow was still there. Once the snow melted, then temperatures warmed, and that's what happened. They waited for almost delay here. 40, 50-day delay to flush in Indian Head, which is another location. So what happens is until snow melt happens, the plants don't produce new leaves. So they are met the chilling needs or vernalization. All they wait is for heat to accumulate. Once they accumulate minimum heat units, they flush, or photothermal units. In poplar, we don't have to use photothermal, just thermal units are enough. Whereas when you reach another tree species, it has to meet minimum photo period as well for leaves to come out. So that's what was going on in those. What we showed was like the low-lying trees were that we made from north and south. When we planted them into common garden, they were all hanging. And when we went after first year growth, we could just see, oh, these are all low. Yeah.
Actually,
We made the process not for biofuel. We made the process to exploit the trait extremes. Once we made the process, the F1s, we started to see very fragile trees. And we realized these are all low lignin types. And when we tested them for lignin, cellulose, hemicellulose, high sugar value. So now we are trying to use this for any biofuel opportunities that we have certain genetics that they can use for breeding.
Temperature, don't.
Yeah, photoperiod is a first cue. I'm not denying temperature at all. Plant realize photoperiod much year to year, they're trained, hardwired. Whereas temperature fluctuate. You can't say what was the temperature.
That is, yeah, photoperiod, but the temperature can drive the process.
Yeah, it is the secondary mechanism. Temperature, water, you name it. Insect, pest are all secondary cues for the plant. The primary cue is day length. Once it understands the day length, then what happens, whether rainfall, good rainfall, it delays flowering. No rainfall, it speeds up flowering. That's it. The mechanism gets speeded up based on the secondary cues. But the primary cue to flower or not to flower is photoperiod. Or how to sense its environment. The plant has to be somewhat ripe, or it has to have minimum number of growth cell cycle. What happens is until that cell cycle happens, you put the seed, it don't produce flowers 5 days later. It waits 35 days, it waits 45 days, it waits 50 days. That is the time it understands, okay, it's safe for me to flower. So it uses day length. Okay, this is the right climate for me to put out flower. When to finish flowering, advance or delay is all next secondary cues. Yeah.
Okay, the minor question is I asked this. So what we are talking right now is totally different than speed breeding. Speed breeding.
Speed breeding, what are you doing?
So we are just manipulating with the photoperiod.
That's all.
Yeah, and then we make them to yearly flower or we get delayed flower.
Yeah.
But in our concept, we keep the photoperiod remains the same, and then we try to whether the plant gets yearly flower or not. So
Yeah.
that's why the difference is and what would the potential, a reason.
In speed breeding, you grow the rice variety for 23 hours and you switch off the light for 1 hour night. What happens is the plant photosynthesizing and it's growing, and the number of cell divisions happen in a day is much higher. The cell divisions keep happening.
So that means we are playing only with reproductive fitness. That's all in speed breeding.
Yeah, that's all you are trying to. Once it reaches a certain, then reproductive switch turnover from vegetative to reproductive switch happens much faster. So the cell cycle, like when we look at a lot of things, the plant don't too many mechanisms. It has kept itself very simple. It don't deal temperature differently, it don't deal drought differently, it don't deal salinity differently. There are certain pathways it uses for any stress, or when insect happens, hormone signals remain the same. It has simplified itself very much. And when we look at humans, when we look at plants, the cells remain the very much the same. How the cells divide, how the cells sense temperature. Our human versus plant, they sense the same way. As humans, we know it is warm and we tell, oh, today is hot, today is cold. Plants sense it. But how they communicate is through neuro signals or releasing certain pheromones or releasing certain chemicals through root signals where each root talks to each other. Mycorrhizal signals all communicate in a different way. So for us, the signal mechanism.
That's cool.
Lot of tree. The question was, not many trees have gone extinct here in India compared to the herbs. I disagree with you when it comes to trees. We have, even in India, as a tropical country, we have lost lot of trees. And also, when you look at forestry building where Bhumi drove me this morning, the biggest office is forest genetics and conservation. So there is a reason for having that. You have to conserve, and certain species are considered endangered. You are not allowed to harvest in forest because the numbers are very low. So that is due to climate change, and some of that are also due to harvesting of wood and other things. So there is a big issue globally. When I go to FAO and all, there is a concern for losing a lot of good genetics or good species that are going extinct around the world. And now there is concentrated effort through FAO to save all those genetics and to understand how these trees behave with climate change and all. Yeah, there were a couple of students in the back.
But on the hydrogen scale regulation.
It has absolute light duration is very important. Intensity will